All right, so we're going to go ahead and look at prokaryotic genomes. This is prokaryotic and eukaryotic genome. You can just cross off eukaryotic genome. It's only about prokaryotes or bacteria cells. And so related to this is a quick picture of a prokaryotic genome. Remember, they don't have nucleuses, so the chromosomes aren't really held anywhere. And they have circular chromosomes. And then they also sometimes have small circular pieces of DNA that are called plasmids. And so they're separate from their main set of chromosomes. So this is their main set of chromosomes, set of DNA, I should write. And then they can also have small circular pieces of DNA. And the first part we're going to talk about is different sources of variation or recombination. How do they get genetic variation into a population or anything like that? So if they're really stressed, they will actually release their genome into their environment, and then they actually can also pick up genetic information from the environment, and that's called transformation. And we're actually going to do a lab where they do that. So they pick up genetic information from their environment, usually in the form of a plasmid, that small circular piece of DNA. In this one, they actually have a small little piece of DNA that gets incorporated into their genome. So one piece gets cut out. They're like, I don't think this one's going to help me survive. So they ditch that one, and then they add in a new one that they picked up from their environment. And then when they replicate, that becomes part of their new genome. So they pick up genetic information from their environment. And we're going to spend a lot of time doing this in our lab, and we're going to make some bacteria glow. So we're going to give them a plasmid that has a gene that can make them glow from a jellyfish. So the gene comes from a jellyfish, and then the little plasmids grow, and it's super cute. Another way they can get genetic information is they can actually send it to another bacteria. So we call that conjugation. Conjugation is basically bacteria sex. Like, they're exchanging genetic information, right? And usually in the form of plasmids. You can see the plasmid replicates, and then it gets sent as we kind of go up this way. So we see that the plasmid then replicates, it gets sent, and now they both have it. So bacteria sharing genetic information. And then the last way is they can do it through viruses. So this is called transduction. And so viruses can contain their own genomes, but then they can also contain viral genomes. And so in this case, this virus infects it, and then it causes the bacteria to actually see right here, there's some bacterial genome going into that virus. So then when the virus bursts or lyse, now this is bacterial DNA. It's not viral DNA. The viral DNA is the stuff that's got more lines on it. So this is bacterial DNA, and it's going to get injected into this bacteria, a different bacteria. And now this is the original bacterial DNA that's way over here, this little piece right here. So we can kind of follow it. I'll just make it all purple for you. So this is all that bacterial DNA getting injected, and now it's part of that. So it's DNA getting transferred through a virus. Okay, and now we're going to talk about operons, and there's two types of operons. There's repressor operons and inducible operons. So the first one is what we call the trip operon, and the trip operon, it makes genes that code for tryptophan. So in the presence of tryptophan, which is down here, we're going to repress it, and in the absence of tryptophan, we're going to turn it on. So in this case, so we have something, so there's different parts. So this right here is what's called the regulatory gene. And it makes a repressor protein. And in this one, it's inactive. This right here is my promoter. So that's that promoter region of DNA. This little piece of DNA is what's called the operator. And then all of this part right there is just the gene. that we're interested in. So they're the genes that code for, in this case, tryptophan, which is an amino acid. So they code for enzymes that help make tryptophan. And so in this one, the repressor protein is inactivated, so the RNA polymerase can bind to the promoter, it can transcribe, and you can see also translate simultaneously, because we're talking about prokaryotes, so there's no nucleus, so the ribosomes can attach, and we're making little enzymes, and we're going to be ready to go. In this scenario, so this is tryptophan, so the tryp is present, so the repressor is activated, and you can see that the tryptophan, or the repressor protein, binds to the operator. So it binds to the operator, and what happens? You guys can see in the picture, the RNA polymerase is literally blocked, and it can no longer, so it can't transcribe the gene, so translation can't happen, and you're not going to make these enzymes that help make tryptophan, which makes sense. Why would I need to make tryptophan if there's tryptophan present in my environment? I wouldn't. What a waste of energy. Why would I do that? So it's a way to regulate gene expression based on what's in their environment and what they actually need, and to not waste energy. Okay. Another classic example of an operon is the lac operon. So same things are happening here. It's just going to be a little bit different scenario. So this is called the regulatory sequence, or the regulatory gene. It codes for my repressor protein, but notice here, in this case, my repressor protein is activated. And so my RNA polymerase can't bind. It can bind to the promoter as it does, but it can't actually transcribe this gene and then therefore translate this gene, because the repressor protein is bound to the operator, so my genes can't be transcribed. Now, these genes are responsible for breaking down lactose. So if there's no lactose around, why would I transcribe and translate those genes? Why would I make a bunch of enzymes that break down lactose when I have no lactose to break down? These genes are also repressed when there's actually glucose around, because glucose is an easier energy substance. So why would I break down lactose if I have an easier energy substance in the form of glucose? Waste of energy. So in this scenario, here's my lactose. My lactose is present. And there is also no glucose present. So then my repressor is inactive. So it doesn't bind to the operator. The RNA polymerase is allowed to do transcription, and then my ribosomes can attach right away and make some proteins, and I can break down all my lactose, and I have plenty of energy in my cell. Okay, so that's a way to regulate gene expression. Only found in prokaryotes. Operons are only found in prokaryotes. We do not have operons. So you are not lactose intolerant because you don't have a lac operon. You are lactose intolerant because of other reasons. It has nothing to do with operons, because you, as a eukaryote, do not have operons. Only prokaryotes have operons. We have other things that regulate our gene expression.